Well, hello, everyone. It's 10 to 2 and it's National Science Week. My name's Doug Record. I am the principal of Boonda Environmental Education Centre, which is a member of the Sapphire Coast Regional Science Hub Sustainability Education Network. And we're gathered here to celebrate science, but also to celebrate 10 years of the Atlas of Life in the Coastal Wilderness. We've got some great people here today to provide grounds for that celebration and some of the achievements that have marked that wonderful 10-year period. I'd just like to, in my role as the Zoom host, because I'll disappear shortly, but I would just like to say, to acknowledge the traditional custodians. I'm here in Boonda National Park on the far south coast of New South Wales between Tathra and Merimbula. This is Diriganj country, part of the Yuin Nation of the South Coast. I extend those acknowledgements to any Aboriginal persons present today. Most people are familiar with Zoom protocols at this stage. If you can have your microphone muted. We chose not to do this as a webinar, which has more limited interactivity, because it is a celebration, and I think if people put questions in the chat, we'd like to be able to see people and to hear from people. But obviously, in a civilized society, we have to do that in a fairly structured way. So don't just grab the microphone and start yelling. If you can put a question in chat or put your hand up or do some of those things, we'll try and give people an opportunity to speak. So I'm going to disappear, but I would like to hand over to our moderator for this event, Annie Lane. So over to you, Annie.
Thanks very much, Doug. Thanks for doing a fantastic job there behind the scenes, or, you know, in front of the scenes in some cases. It's terrific. Good afternoon, everyone. I am the chair of Budawang Coast Atlas of Life, which is the sister project to the north of Atlas of Life in the coastal wilderness. So as Doug mentioned, we have two speakers in this session, Libby Hepburn and Thomas Mesaglio. Now I will start with Libby will take us off, and Libby is probably one of the most passionate and committed people that I know when it comes to citizen science activity. She's been instrumental in putting together this really interesting program that we have this week during Science Week. And she's one of the founders of the Atlas of Life in the Coastal Wilderness. So Libby, as a part of a celebration of the 10-year history of that project, will be taking us through what the journey's been like and some of the milestones along that journey. Libby will then hand over to Thomas. And Thomas, thank you very much for joining us. Thomas is a researcher at the University of New South Wales. He is incredibly active also in the citizen science space, and he is passionate about all areas of natural science, particularly marine science. He is the curator and moderator on iNATURALIST, which is the citizen science platform of choice for many, many citizen science projects. And we just heard about one being the bee hotels. And Thomas has been fantastic in helping both of our projects to move over to this platform. So Thomas is going to share with us some of the ins and outs of iNATURALIST and how we can best make use of this really fantastic program. And Thomas is also an organiser for the Great Southern BioBlitz, which will take place in later October. And so Thomas is going to tell us how we can get involved in that project. All right, Libby, I might hand over to you. And as Doug mentioned, I encourage you to send in your questions via chat or Q&A during the presentations, and we'll get to those at the end. Thank you. Thanks, Libby.
Thanks very much, Annie. I'll see if I can share the screen now and make things happen. I'm just going to do a quick run through of the Atlas of Life first 10 years and tell you a bit about what it is and why we started it. I'm hunting through the archives, I discovered this, and I knew it was somewhere around our 10-year anniversary, but it's actually the 10-year anniversary is Friday, the 19th of August. So we couldn't have got much closer than that, could we? And we started off, Donald Hoburn came down to Merimbula and launched the project for us because the Atlas of Living Australia wanted to get communities involved in using the platform. So my PowerPoint seems to be messing about a bit, so I'll apologize in advance. So why did we do this? We did this because a group of us really keen naturalists are really worried about kids growing up with that sort of life instead of this sort of life. So one of the main reasons that we have for setting up the Atlas was really to provide opportunities for people to get closer to nature. And we were also, there were quite a lot of things going on that we were interested in and wanted to know more about. We were told by a leading expert that we might lose all the giant kelp from the whole of Australia within 20 years. We know that things like the channel-billed cuckoos are changing their ranges, and similarly with flannel flowers. We know there are invasive species like the European green shore crab. So we thought that if we could record what's out there, then we'd have a chance of knowing, learning more about it and working with scientists. And we also recognize that we need to bear witness to some of the things that are happening. Now, this is one of the superb eastern curlews that we believe will be extinct within whenever its lifetime finishes. And we get these on Merimbula Lake, so we want to record these and know what's happening. So lots of reasons why we decided to do this. We want to encourage people to spend time in nature. We want people to record what they see, build a community of practice with individual and group expertise. We want to work in partnership with natural resource managers and scientists. And we also want to encourage new generations of taxonomists. So our work is to remind people how wonderful nature is and our environment. And we're so lucky to live here. So we have lots of opportunities of creating events and activities for people to get involved with. And I found this as well. So we've been doing Science Festival for quite a while now. I don't know if this was the first one, but certainly in 2014, we were already at it. One of the things we did to celebrate science was to set up a symposium of the history of science in our region. And that was fascinating. We got some brilliant world-class ecologists here. This photograph here is from 1974, collaring dingoes in Naji. And the wonderful Peter Fulliger, who works on Montague Island. At the time we did this in 2012, he had been researching over the last 50 years, regularly every year. So now it must be nearly 60 years if he's still doing it. So that was a fantastic, we got 90 people at the symposium over 2 days, and it was wonderful. And we've done things like help to set up identification boards at the blue pools so people can see some of the wonderful things they do. And obviously our target, a lot of our target is to try and encourage children to get into nature. And with that, we've got one of our great partners is Doug, who at Bournemouth Environmental Education Centre does wonderful things, and we work with him as much as we can. One of the big ticket items that we decided we would use to promote people getting into nature is the BioBlitz, which is a thing where you get lots of scientists, lots of naturalists, lots of community, and you record as many different bits of biodiversity as you can. And the first one was in Bermagui in 2012. And we've got the t-shirt on here from that one. Still keep it. It's one of my proudest things. We've done 7 in this area, all sorts of different reasons. This one we were working with the Bega Land Council, who'd they got some land given back to them, and they wanted a baseline data. And we've done all kinds of different things. This was at the Mimosa Rocks BioBlitz, which was our most successful in terms of numbers, where in 2 days we got 1,008 different species recorded. And this was the base camp we had, and these are some of our amazing volunteers, Jenny and Graham Greenwood, and the calendar that we did in 2015. And it's not just the kids who like to get down and dirty. We have wonderful times with adults of all ages doing various things, learning different things. And this again was at the Mimosa Rocks BioBlitz. And one of the surveys that we had was, this is Chris Allen, who's been researching the remnant populations of koalas in this area. And they ran, I think, 12 surveys over the 2 days. And on the last day, if you look bottom right here, you can just see by the expressions on everybody's faces, they actually found some fresh koala scat. And they were so chuffed. They were absolutely delighted. It was a wonderful thing. And again, this was at the Four Winds BioBlitz, where they were wanting to create an arboretum. And we did a baseline BioBlitz for them at the site. And one of the people, Helen Ransom, found a spider under the guidance of the survey leader, Stuart Harris, who was amazing. And it was a new species. And he very wonderfully took on the job of getting it, whatever you have to do with it to get it to become a species. And he named it Muratus sapphirus. So it's the peacock spider of the Sapphire Coast, which we're all very proud of. So you do get wonderful things from BioBlitzes. And this was our website that we had for a long number of years. And it's now, looking back on it now, it looks a bit clunky, but it did very well in terms of what we needed at the time. And one of the things that we did every year, we've run photography competitions, because we believe people get inspired by the beauty of nature as much as anything else, as much as the science. And photography competitions are wonderful ways to get people to bring fantastic images to us. And we've had some brilliant images and brilliant photographers. And all of these are on the archives of our website. So if anybody's interested, they can see them. We've done lots of celebrations of various things. And this one, we got Joe Baker was the chief scientist of Queensland, who came down and did a presentation to us about seaweeds. And Alan Miller was the chief phycologist of Sydney Botanic Gardens. And newsletters. This newsletter, I had a look at it, 16 pages it was. So there was a lot of work to do, but wonderful things that we found and we shared. And we invited lots of different experts who really enjoy coming and talking to people down here. In fact, we found that the people who are passionate about their subjects are the most generous with their time, and they love talking to people who are interested in what they're doing. So it's actually a real feedback loop with the naturalists and the scientists that we work with, which is really great. And we've built wonderful relationships with all sorts of different people in the area that we've worked with, and we continue to work with over the years. And we've had a gong. There you are, Fiona, there's you and me collecting the gong. We got a gong at the Banksia Community Award in 2018, which is great. It says something about what we're trying to do here, that we're trying to support community and science becoming partners in things. And it was really nice to do that. And we've done work with the Bega Valley Shire Council. This is we have a very rare plant, which is Marimbula starhair, and we've helped them to locate it in the area. And we've also gone on from that, and we've had people coming down, and we've working out what the pollinators of this plant are, with a view to that helping to determine the management management regime for it. And that's always interested people. Saving our species, we've been involved with, and some projects we haven't got as much engagement as we would have liked, but we can do something about that. Beach weeds, printed 1,000 stickers to stick around the area so that people could help the council to record the weeds that are happening on the beach. And one of the most successful ones has been our engagement with the sea slug census, which we've been doing for 4 or 5 years now. And people love it. And some of the research papers that are coming out of it in terms of climate change in the ocean are really fascinating. So that's something that we'll work on. And a few years ago, a lady called Kerry Lee Harris took over as chair of the Atlas. And she did amazing things. She's a scientist from Melbourne, and she retired with her husband, Paul Whitington, to Womboyne. And she got involved with the Atlas and got very interested in it. And she created a new website for us, which took us into another sphere. And she did wonderful, sophisticated things with it. They're great photographers, and they produced lots and lots of papers and things, information for people to dive deeper into the species we were looking at. And she set up Atlas Naturalists again, which we'd become fairly relaxed about, and set up events and things that she took people, and people got very enthusiastic about it. But then we had the bushfires, and it was terribly sad because their beautiful house at Womboyne, everything around the house was destroyed. And these are just 2 little bandicoots that they'd survived the fires, but there was nothing to eat. So that happened. And unfortunately, Kerry had to deal with all of that. So she stood down. So we carried on with the Atlas because there's lots of things we can do. And this year we've signed a memorandum of understanding with Bega Valley Shire Council, which is fantastic, which means we're going to work as partners with the council on at least 4 projects every year. And that to us, after 10 years, is a really important milestone that we've achieved. And one of the things we've set up, because we want to promote iNaturalist in communities, is with the council, we've set up a project which is to look at life in our cemeteries, in and around the cemeteries, all the cemeteries in Bega Valley Shire. We're going to look at over the next 3 years, and we were going to have a BioBlitz there on next Saturday as part of Science Week, but unfortunately that's had to be put forward a bit. But still, it'll come, we'll do it, and it'll be great to get out with the communities. And this year we've transitioned. We've had a couple of recording platforms before we got to iNaturalist. The first one was the Atlas of Living Australia's own platform, which was, I mean, that you have to start with something, and it was a good start, but really quite difficult. And then we had Nature Mapper, and then we had to move from Nature Mapper this year, and we've moved over to iNaturalist. And I haven't got time in this very short presentation to thank all the people that have been involved with us, but I would just like to say thank you very much to the lady with the computer down here, is Patricia Daly, who's been our database manager and supporter and a huge resource for us all the time that we've been working together. This was at the Pambula BioBlitz in the middle of the night when she was helping people record things. She's a wonder. But now we are moved over to iNaturalist. We now have a platform that everybody is really enjoying. Everybody's finding it really easy to use, and the app is reliable. So it's a really good thing that we have here. And we're delighted to have something we can go out and promote to the whole community again. And we hope we can do lots of work with the Budawan Coast. It's great to have a sister organization who's trying to do the same things as we are. And we've got several projects that we want to work on. The wonderful Southern Hemisphere Great Southern BioBlitz, which is going to happen in October, is a fantastic thing for us. And we'll be promoting it a lot. To see if we can get lots and lots of people involved. That's going to be a real goodie. And tomorrow we've got Michael Mulvaney talking about his project, he's wanting to extend the research on gangang nesting through the whole of the Atlas of Life, coastal wilderness and Bedouin coast areas. And he needs a bit of help here, Thomas, because he needs some help getting that photograph right, because it doesn't work. But that's a nice segue into what's happening next, because one of the big things that we needed to do when we transitioned to iNaturalist was to learn about the platform and what it could do. And Thomas has given, he's been hugely generous with his time, and our moderators and our contributors have learned a lot from what he's had to offer us. And we hope that will continue over the future. So there's a very quick snapshot of what we've been doing for the last 10 years. And now I'd like to hand over to Thomas to deep dive into iNaturalist. If I can get out of this, that is. I've stopped sharing, there you go.
Great, thank you very much, Libby. So pretty much how I was going to run today was, whenever I do these kind of workshops and presentations, I tend to prefer to have as few slides as possible. And I prefer instead spend most of my time in iNaturalist itself, just so I feel like it's a lot easier for people to follow along if I'm actively doing the things that I'm talking about. So I'll pretty much just run through 7 or 8 slides for say 5 or 10 minutes, giving a bit of a brief overview of iNaturalist. And then I'll dive straight into the platform itself and delve into a few kind of tips and tricks for how to use the platform and how to really maximize your usage of it. And I will just highlight that most of the stuff I'm doing today is pitched at quite a basic level in terms of iNaturalist usage. So I apologize for people that are a little bit more advanced with the platform. A lot of the stuff I'll say you'll probably know already, but I will highlight that on the 7th of September, I will be running an advanced iNaturalist workshop. So anyone who's keen for some real tricky things that you can do with the platform, you're more than welcome to attend that. So I will dive into my PowerPoint. One second. Okay, awesome. So for people who aren't aware of the platform at all or have never used it, what is iNaturalist? It's essentially an online biodiversity citizen science platform. And what I mean by that is it's essentially a way for people who aren't necessarily professional scientists to be able to collect data and interact with real life scientists and then have said data be used in real life research. So it works in a way that you go outside or even inside, there's plenty of life that you'll be surprised at what you'll find in your house. But you go out and you take a photo of something or you take a sound recording and you upload that observation to iNaturalist, and then other users from all around the world, both amateur naturalists and many, many experts, are able to identify those observations for you. And then once your observations have been identified, the data get exported to databases such as the Atlas of Living Australia, just from a national perspective, but also the Global Biodiversity Information Facility, which is basically like the international version of the ALA. And from there, of course, scientists from all around the world are able to use your data in real life research. Now, as of yesterday, just as a bit of a highlight of exactly how successful iNaturalist has been, you can see that there are almost 77 million observations that have been uploaded by almost 2 million observers. And this is stemming from 2008 onwards, from when iNaturalist launched. So in just over 10 years, we've seen almost 80 million observations across 344,000 species. So this growth has certainly been reflected from an Australian perspective as well. And if you look at just Australian statistics, you'll see that we're getting very close to 2.5 million, contributed by almost 35,000 observers. So if you look at a very brief history, and these are just 2 figures I pulled out from a recent paper I published about iNaturalist in Australia. you'll see that in the first, say, 6 or 7 years, things were pretty slow. And that's understandable because iNaturalist, to start off with, was largely a US-centric platform, having started in California. So really, for the first 6 or 7 years, we didn't really see much action from an Australian perspective. But then you'll see that from around 2016, the middle of 2016, early 2017, you have this sudden little jump, which really started to spark observations. And what that jump was the Australasian Fishes Project launching, which some of you, a lot of you might be familiar with. And that's one thing I'll highlight as well in my presentation, the importance of projects. But you see, pretty much from there, we never looked back. And ever since then, both number of observations and research grade observations, as well as the number of species and the number of observers, have all been increasing exponentially for Australia. And indeed, at the start of this year and the end of last year, Australia exceeded 100,000 observations uploaded in a single month. So this is another figure I pulled out from my paper, just to give you a broad sense of where observations are being uploaded in Australia. So this is a relative density heat map. So the hotter the colors, the more observations have been uploaded. And immediately you can see that there are some obvious biases towards the East Coast and Southeastern Australia, as well as obviously all the major capital cities, with pretty much nothing in the interior. So if you are planning on doing some road trips whenever this pandemic ends, this map is a good place to try and plan out where you should go to try and fill in some of these gaps on the map. And one question I get asked a lot when I'm talking about iNaturalist is, and this is from an expert perspective, is why should I join? So a lot of my research around iNaturalist revolves around trying to increase the amount of data that gets uploaded to the site, to increase the quality of the data, and also to recruit more experts. So one of the things that we've found quite a lot is that when experts join iNaturalist, they have this incredibly disproportionate impact on the quality of the data and the way that people interact with the site. And I'm sure all of you that have used iNaturalist can think of examples off the top of your head. One person who some of you may be aware of, either from iNaturalist or from real life, is Kevin Bonham. He's a land snail expert from Tasmania. And if you've ever uploaded any land snail observations to iNaturalist, he's probably ID'd them for you. And he's ID'd something like 75% of all land snail observations for Australia in iNaturalist. So that's a single person. Another example exactly the same is Lee Windsor, another guy, another name that a lot of you might be familiar with. He's an international and Australian expert on flatworms, terrestrial flatworms. And again, he's ID'd something like almost 3/4 of all flatworm observations uploaded to Australia. And this is this really cool snowball effect that this ends up with, where you have more experts that get recruited to iNaturalist and start contributing their expertise and their identifications. With their identifications, you get this great process of the amateur naturalists and the true citizen scientists being taught by these experts. In turn, because of their greater knowledge, the citizen scientists make better identifications themselves and also become motivated to make more and more observations and more high quality observations. The data improves on iNaturalist in turn, which then acts as a greater incentive for more experts to join. And you get this amazing cycle where more and more experts join, the data gets better and better, and you just take off from there. So it highlights this really, really important feedback loop where iNaturalist is just this really amazing platform for the dissemination of knowledge from the experts to the amateur naturalists. And finally, just in terms of the actual research output, so this is another really common question I get with, when I upload data to iNaturalist, does it actually get used in real life research? And essentially it boils down to, a lot of people are interested in, will my data and observations actually be useful? And the answer to that is a resounding yes. So especially in the past year or two, more and more papers and research are using iNaturalist data. And indeed, there are probably hundreds of papers now in scientific journals that use iNaturalist data, from absolutely everything from rediscovering lost species that were thought to be extinct, to understanding how our ecosystems recover from natural disasters, even to tracking population trends of entire large taxonomic groups across a continental and decadal scale. right? So all you have to do is jump onto Google Scholar and plug in iNaturalist, and you'll see literally hundreds of papers that are using iNaturalist data. And indeed, many of those papers probably use observations that you guys have uploaded yourselves, which is, I think, a really, really cool and fulfilling thing. So that's enough for the presentation itself. I'm going to dive straight into my actual use of the platform. So pretty much I'm going to go through kind of 6 main things for the platform. I'm going to first delve straight into making observations. So obviously observations are the basic unit of iNaturalist. So I'm going to quickly race through how to make observations and what observations actually entail in terms of their metadata. I'll quickly go through the Explore tab, which is where you can basically explore all observations on iNaturalist and set up some very nice filters and pretty much look at the areas you want and the taxonomic groups that you want. I'll go through the Identify tab, which of course allows you to then identify observations for other people. I'll look at very briefly subscriptions and notifications, as well as then rounding out the presentation with looking at how communication is one of the most effective parts of iNaturalist and how the social aspect of iNaturalist is one of the most important aspects of the platform. But in terms of observations, and also keeping in mind that obviously this presentation will be geared towards the desktop version. Of course, there is an app version of iNaturalist, which is really, really great if you're out in the field. But as I always emphasize in all of my presentations, the apps have greatly reduced functionality to the desktop. So if you are out in the field and on the go, by all means, use the app. And it is fantastic as a portable version of iNaturalist. But if you are at home and you do have your computer handy, I highly recommend using the desktop version just because there is so much more functionality and way, way more features. So with that in mind, if you are looking to upload observations, all you got to do is click the big green button in the top right corner, upload, and you'll get taken to this screen. And then you've got 2 options. So one of the options you can do is you can click choose files, and that'll then actually take you to folders in your computer. But if you don't want to do that, you can actually just drag and drop straight from those folders as well. So if I bring up this, I can actually just drag files straight onto my, my upload screen. And if you want to do multiple ones at once, you just click on the first one, hold down the Shift key, and that'll get everyone in between. And then you can drag those all at once, and you'll see they all start loading. I'm just going to remove those for now. But let's say I just want to upload these 4. So we've got our observations here, and you'll see that in most cases, the date has already been filled out and the location has been filled out. So if you've used a phone to take your photo, then in most cases, the location will already be filled in for you. But let's say that you have taken the photo with a camera and your camera didn't have a GPS on it, or you did take it with your phone, but the GPS was wrong for whatever reason. All you have to do to change the location is click in the bar, and then you can actually just physically reposition. Okay, so all you got to do is click on the map and you can physically place where your observation was. So let's say my observation was right here. You'll notice that there's a little red circle around your observation. And what that red circle is, it's the accuracy circle. And what that does is it indicates that you're not 100% sure where your observation was, but it was 100% in that circle. So if I, let's say, was doing a walk through this reserve, this is Watagora Reserve in South Granville in Sydney. And let's say I remember that I took a photo at some point in this area here, but I wasn't sure exactly where. All I have to do is grab one of those nodes on the edge of the circle, cover all the areas where I know, press update, and that now indicates that I saw that plant somewhere in that circle. So that's a really good way of indicating uncertainty in terms of your location accuracy. One other thing you'll notice with the location is this little thing called geo-privacy. And you'll notice there are 3 options: open, obscured, and private. And what these mean, so if you set yours to open, which is the default option, it means that anyone can see the coordinates of your observation. So the observation data in terms of location is completely open. If I set it to obscured, What this does is it will randomly scramble your true coordinates into a roughly 0.2 by 0.2 degree box, which equates to 500 square kilometers at the equator, around your true coordinates. So it will randomly scramble your coordinates into a big box around your true coordinates. And the reason this is a really useful feature is it means that it protects, or it helps to protect, threatened species. So let's say you go out and you find a rare plant that is often a rare orchid that is often threatened by collectors. And you really want to upload this observation, of course, and add to scientific data on the species, but you don't want people to be able to look up your observation, say, okay, Thomas saw this orchid at this exact spot. I'm then going to go back to that exact spot and find the orchid and collect it. So obscuration is this really great way of combating that, or at least one way. Any species that has a conservation status of near threatened or above will automatically have its location obscured. So you can both choose to obscure things if you want, but keeping in mind, if you upload anything that is endangered or threatened or vulnerable, iNaturalist will automatically obscure the location for you. So you don't have to do anything there. The final option is private. This is one that I generally will advise against using. The reason for that is if you set your observation to private, it will completely remove all location data. So anyone else looking at your observation won't even know what continent your observation was made in. That makes it a lot harder for people to find your observation and to identify it for you, because when they search, a lot of people will say, search for New South Wales observations of butterflies. But even if your observation was in New South Wales and it was a butterfly, if you've set it to private, it will not appear in that search. So I only recommend using private if there is an extreme risk of collection. So these are very few rare cases, things like very rare snakes in North America, or there are a few very rare orchids and cacti across South America, for example, where even obscuring the location still does not provide sufficient protection. And so some people will set them to entirely private. So we can just go open for now and go update. One other thing to notice on the upload screen is that you have this little bar where you can type in what you think your observation is. Now, there's two important things here. The first is that always, always, always enter an observation ID, even if you have no idea what it is. So even if it's something as basic as plant or animal or fungi, that is completely fine. So one common pitfall that a lot of new users will fall into is that they will upload an observation and they'll leave it completely blank in terms of the ID. Again, this makes it a lot harder for people to find your observations and help identify them for you, because the way that many people ID things on iNaturalist is by searching. So for example, one thing I often do is I will search for mollusks in Australia to identify. But if you have uploaded a mollusk but you didn't add an ID of mollusk, then it becomes invisible to me through that search. So even if you have absolutely no idea what kind of shell you've just found on the beach, adding an ID of mollusk means it will appear in searches for mollusks and it will become a lot easier to ID. So in this case, even if I had no idea what this was, all I have to do is say plants. And now anyone searching for plants, which is a lot of people in iNaturalist, will be able to easily find your observation. But let's say I want to use the suggestions. So one of the really cool features about iNaturalist is that there are computer vision suggestions. And these are the ones that appear when you click in the bar, and then you'll see the drop-down menu. So this is basically the artificial intelligence software that iNaturalist uses. So this is a really amazing feature that was introduced in 2017. You'll probably have seen features like this before. Google Lens is another very famous one, where essentially the AI looks at your photo and it tries to guess what it thinks the species is. And these guesses are informed by a big set of training data. So every time you upload an observation and you identify it and it goes to research grade, then the next time the computer vision gets trained, it will have your photo in its training set. So basically every time you identify something and it goes to research grade, you are telling the artificial intelligence, this is what a magpie looks like, or this is what a meadow argus looks like, or this is what a spear thistle looks like. And the computer vision keeps learning and learning and gets better and better at recognizing what species look like from all kinds of different angles. So you'll see we've got this photo here that I took, and you'll click in the bar and you'll see that the option is spear thistle. So this is quite an easy one for the computer vision because spear thistles are a very, very frequently observed weed. And so it's got really, really good at figuring out what it is. So then we can test it on this one. So this one is a bit of a tricky one. So this one is a water gum, Tristaniopsis lorena, which grows along watercourses in New South Wales. So we'll see if the computer vision can recognize this one. And it does. So indeed, it is one of the top options. But you'll see now that more options have appeared. So it has become more uncertain. And then finally, test this one as well. This is native spinach. And you'll see that again, it's the top option and it recognizes it straight away, but there are, again, more options. So what this is basically saying is that there are more species that look quite similar to this one, that it could possibly be these species as well. Whereas here with the spear thistle, it is quite certain that this is what the species is. Now, one thing to keep in mind, so whilst the computer vision is really great with observations that have, well, I should say species that have lots and lots of observations and really easy things to ID like birds or really common weeds, when you start feeding it things like obscure insects, it's not so great. And that's understandable because, of course, most of the observations are from North America because that's where all the observations are. So the training data is based on those observations, which means a lot of the suggestions it gives you are of Northern Hemisphere species. So it's always important that if you are going to use these computer vision suggestions, that you check the distributions first. So I could click here and it tells me it could be one of these 3 different species. A good option, if I'm unsure, is to just click view. And what that'll do is it'll open a new tab and it will give me some extra photos of this species. It'll tell me exactly where this species has been seen. So I can say, okay, most of its distribution seems to be along the eastern coast of New South Wales. It seems like a pretty good mix, a pretty good match, I should say. And that should just give you at least a little bit more certainty over whether you should pick that observation or not. You know, and the obverse is true. If you pick one of those options to check out and you see that every single observation is in China or in South Africa or in the US, you can say, okay, maybe it's not that species. Maybe I should go to a coarser level to be safe. That pretty much covers everything in terms of making observations, because I'm aware that I've got very limited time and I've got to whiz through things. But one last thing for particularly very new users in particular is that each observation equals 1 encounter with 1 species. So that's one of the most common things I'll see with new users uploading observations is they'll do one of 2 things. So one circumstance is where they'll take 4 photos of a bird and then upload those as 4 separate observations. Whenever you encounter 1 species at one time, they should all of your photos of that species should be combined into a single observation. And the way you can do that is by dragging and dropping the photos in this screen. So let's just pretend that all 4 of these photos are of the same individual plant that I saw. All you have to do is pick up each one and physically drag it. And all of those photos will be combined into 1 observation. You can see there it says 1 of 4. And then you can use the arrows and then you can see those all. And the one that you drag the observations onto is the one that will be the first photo to be shown. So because I dragged all the observations onto this photo, this is the one that will show up first. On the flip side of the coin, you should not have multiple photos of multiple species in the 1 observation. So a lot of times I'll see a new user upload an observation and it'll have 3 photos in it. And 1 photo is of a bird, 1 photo is of a mushroom, and 1 photo is of a plant. So each of those ones should be in their own separate observation. So that is pretty much all I have for the real basics of uploading observations. I mentioned that advanced workshop that I'll be delving into. I've also got a few beginner workshops teed up. One is tomorrow night, so you're more than welcome to join that. And I'll pretty much be going over the same stuff, but in a lot more detail. So if you want to delve a lot more into the mechanics and the tips and tricks of iNaturalist from a basic perspective, I have got a workshop going on tomorrow night that I can send a link to Annie and Libby and Fiona to send out to everyone. So that's how to upload observations. But then let's say you want to explore things and you want to look at observations made by other people, or potentially you want to try and identify your own observations. Yes, Erica.
Hello, hi, sorry, that was very quick. I wasn't expecting to have to ask my question immediately. Just a quick question about the multiple species in one observation.
Yep.
I have a project set up, which Michael Mulvaney is actually co-opting for some of the gang work. And the purpose is to get records of parrots and also their food species. And we've been trying to work out the best way to get both things identified without causing confusion. Because obviously uploading the same photo twice will get confusing if one is supposed to be for the bird and one is for the plant.
Yeah, so that's completely fine, uploading the same photo. So, and you can actually do it in a really fast way. So
just so we can tell people the best what to do, really.
Yeah, no, 100%. So let's just say that I have uploaded the bird photo first. So let's just go through some of my bird observations.
I mean, you can go to the project if you want. There's examples there.
Yeah. What, in the hungry parrots or the
yeah, it's the hungry parrots one. I'm trying to whip it into shape as well.
So I will just go with one of mine as well, just because the feature will only be available for mine.
Yeah, who are you on iNat, by the way? I wondered if that was you. Excellent.
Yes. So what you will want to do, and I'll jump to the project in a second, but let's say that I have uploaded this photo of a bird, right? And I've got it and I've ID'd it as a bird and everyone's agreed. But in it, I'm really interested in what the tree is. Now, obviously in this case, it would be quite tricky to narrow it down, but all you have to do, and this is what you can tell people to do, is you go to the observation, click the down arrow next to the edit button, and then click duplicate. And this copies every single aspect of the observation, including the photo, including all the metadata. And now all I have to do, and I'm cheating a bit here because I know what the species was.
But that's fine. If you know what it is, then that's all good.
Yeah, so all I have to do is do that. But let's say I don't know what the species is. Then all we've got to do is go with flowering plants.
Yeah.
And then we go save observation, and that's pretty much like the suggested way to go through things.
As project admins or curators or whatever we're called, are we able to do the duplicating ourselves? So if someone's only uploaded it once, yeah, no, okay. No. So we've got to ask them to do that.
Pretty much, yeah. So unfortunately, it is a manual thing that does require people actually responding to your requests, which doesn't always happen. But certainly that is the recommended way to go about doing these kind of things. And
yeah, okay, cool. And would it be good to add into the caption, this identification is for the bird as opposed to this observation is for the plant?
Yeah, 100%.
So people don't 100%. Then go, what are you talking about? That's not a eucalypt, that's an Eastern Rosella, you idiot.
Yeah, no, 100%. And that's a really common thing you get. So to explain what Erica's saying there, if you do have a situation where you've got two possible subjects, or perhaps one subject is less obvious that you want to focus on, all you have to do is click that edit button again. And here you'll see there's a notes section on every one of your observations. And all you have to do is go, this observation is the plant. Or I would actually recommend, in a case where it's not so obvious like this, I would actually recommend doing that as a comment.
Yeah. Okay.
Because I find that more often than not, people will read comments on an observation than the description.
Yeah.
So, it's not the best solution, unfortunately, in terms of getting the focus, but certainly iNat's policy is quite clear in that one observation equals one species. So the fastest and the cleanest way to do things is just to duplicate like that. And it certainly does save time as well, instead of making you re-upload the observation all over again.
I believe you can link observations too, or is that an advanced?
Yeah, you can. So I'll probably leave that for a more advanced workshop.
Yeah.
But certainly you can, and I'm happy if anyone here is keen about doing that. I'm happy to chat and you can message me afterwards. But you can then set up a way to link observations where you then click on the link and it will then show you all observations in that set. So a really common way that people do that kind of thing is if they're tracking, say, caterpillars through their life cycle, they'll have an observation for the caterpillar, and then maybe in a few weeks, they'll then make an observation for the pupa. And then in a few weeks, when it hatches, they'll make it for the adult. And then you can link all 3 observations and you just go to one link and then you can see all 3 at the same time. So certainly
cool.
I can show you how to do that, Erica, afterwards.
Thank you.
No worries. So one thing that I mentioned was the Explore tab. And this is a really great way to look through other people's observations. So let's say that you're really interested in looking at butterflies in New South Wales. All you got to do is go to the search bar, type in butterflies. And I am keeping in mind that there are 6 comments in the chat, so I will get to those questions afterwards, don't worry. So you search for butterflies, and then you search for New South Wales. And away you go. And you'll see that so far for New South Wales, just over 11,000 observations have been uploaded for butterflies. So if you look on the observations tab, this is a way to manually scroll through every single observation and look at them. And all you've got to do is click on each one to actually look at that observation. What I'd recommend if you're doing it here is actually to right click and open link in new tab. That way you'll have both this page still open as well as the individual observation you can go to. Now, when you're doing it this way, each page will show 96 observations. So if I scroll all the way to the bottom, and this is a little tedious, you'll see that you can then go to all the different pages. So each one individual page will have 96 observations on it. What you can also do is go to the species tab. And so let's say you're interested in individual species. So let's say I really wanted to look at a meadow argus observations in New South Wales, and I didn't want to have to scroll through 11,000 different observations to see them all. All I have to do is go to the species tab, and then you click on this link. So make sure you don't click on the species name because you'll see what happens there. You'll actually go to the broad page for meadow argus. So instead, I'm just interested in the ones in New South Wales. So instead, you'll click on this observations link. And now I'm looking at all the observations of meadow argus in New South Wales. Nice and easy. The other tabs you'll notice here are identifiers. So you can see who the top identifiers are of butterflies in New South Wales. So Peter Ewan is someone you may have encountered. He's a real whiz with butterflies and a great guy to ask about IDs. And you can see pretty much all the people who have ID'd. And then you can also look at the top observers, and that gets ranked by both the number of observations and the number of species that they've seen. So this is a really, really great way to get your things identified. So as one example to illustrate my point, let's say, and we'll get a good example here for one of my observations. Okay, let's say we've got this jumping spider. So here's a jumping spider that I saw 5 or 6 days ago, and I'm unsure what it is. So, and I want to get it identified. So how can I do that? First, I'll go to the Explore page. And what I'll do is I will type in jumping spiders, and then I'll narrow it down to New South Wales because it's probably not much use bothering people who identify jumping spiders in the US because they probably won't know the local New South Wales species. So now I'm looking at jumping spiders in New South Wales, and I've got 2 different options here. One is that I can compare my photo with other photos. So what I can do is I can say, okay, I've got a good sense of what this photo looks like. I can go to the species tab and I can start scrolling through all these thumbnails and I can start comparing. And it's quite easy to cross things off, of course. You can say, okay, it's obviously not this, obviously not this, obviously not this, and then progress my way through. But obviously this isn't a foolproof way of doing things because many of these spiders are sexually dimorphic. So the males will look quite different to the females. And so obviously you've only got one little thumbnail image here for each. Of course, you also have the possibility that mine is a juvenile, in which case it might not be hugely useful to just scroll through here as well. So my next option is to say, okay, maybe I should look at one of the top experts at jumping spiders for New South Wales. All I have to do is go to the identifiers tab, and I can see who the top identifiers are of this group. And straight away you see that one person, two people, but in particular one person jumps out. And this is a user called Susie and Jim. And from experience, I know they're really great with identifying jumping spiders. So what I'll do is I'll go back to my observation. And now, just like you would tag someone in any form of social media, I'll type in the @ symbol, and then I'll type in their username. And you'll see that there's an autocomplete as well there for you if you can't quite remember their username. And I can tag them. I can say what I think the genus might be, and they will now get a notification. And the notifications are up here in this red little speech bubble. So whenever you have a notification, it will appear as a red speech bubble. And this is one of the most important parts of iNaturalist. So again, with the social media analogy, just how you're on Facebook, Instagram, Twitter, every time someone mentions you in a comment or tags you in something or sends you a message, you'll get a notification. The exact same thing happens on iNaturalist. So every time someone tags me in an observation, or they make a comment on an observation that I've interacted with previously, or someone identifies something for me, it will leave a notification here. So it's really important for you to check your notifications every day, because they're the main way that other users interact with you and help you update your content. So whenever Susie and Jim get back to me and potentially, hopefully, fingers crossed, identify this spider for me, I will get another notification that will bump up to 5. Now, as well as making observations, and I'm aware that I'm rapidly running out of time here and I want to leave some time to answer people's questions. So I'll just quickly dive into 2 more things. The first is the Identify tab. So we've covered observing things, right? But the other main part of iNaturalist is actually identifying things for people. And of course, you know, everyone here is able to offer identifications for other users. And the way to do that is through the Identification tab. Now you see that it's pretty much the same setup as the Explore tab, but a little bit different. So by default, straight away, you'll see if you go to these filters, only needs ID things turn up. So these are ones that haven't been confirmed yet. And just to clarify what needs ID means, when you have an observation get uploaded, it is first of all labeled as needs ID. But then once people start adding identifications to that observation, it can eventually reach research grade. And the way things reach research grade is if they have at least 2 observations and more than 2/3 of those IDs, I should say, have to agree. So if I add an observation, it just has 1 identification on it, it will be needs ID. If 2 people have identified an observation and both of them agree, it will become research grade, because 2 out of 2 is more than 2/3. But let's say a 3rd person comes along and they add a different identification that disagrees with those first 2 people, it will now get bumped back to needs ID, because 2 out of 3 people agree, which is not greater than 2 out of 3, so it is needs ID. So the way to get things to research grade is to have 2 out of 2 IDs agreeing, or 3 out of 3, or 3 out of 4, or 4 out of 5, or 5 out of 6, and so on and so forth. And obviously, the more identifications that get added that are all disagreeing with each other, the harder and harder it gets to become research grade. And this is iNaturalist's way of creating this community consensus. So if there's a lot of disagreement over the identification of something, then it will remain at needs ID until some kind of agreement is reached. So as I said, this is a great way to identify things for people. So let's say again, we'll filter it to New South Wales butterflies. And in fact, I'll use mollusks as an example here, just because there'll be some things that won't have been identified yet. So you'll see straight away there's this handy little agree button on a lot of observations here. So if you agree with an observation, and you can click on the thumbnail to make it bigger, all you have to do is click that agree button, and you have now identified something for someone. So you'll see that the observation now becomes faded, and it's has this little thing here that says reviewed with a blue tick. And it means that I have reviewed this observation and I have identified it for someone. However, you'll see that for other ones that aren't identified as species, there is no agree button. So all you have to do, so let's say I recognize this one. I know that this is Patelota altocostata. So all I have to do is click the add ID button, I will start typing in my ID. I click it, I click save, and I've now added an identification for someone. So it's as easy as that. So if you're wanting to identify things for people, this is the screen that I highly recommend. You can upload things, you can identify things, I should say, for other people through the Explore tab as well, but you'll see it's a lot more tedious to do. So if I was to try and identify things for people this way, if I click on this to go and identify it for them, sure, I can do that, but then I have to click back and go all the way back to this screen, or it forces me to constantly open things in new tabs all the time. So this is a very tedious and time-consuming way of identifying things for other people. Whereas if you just go to the Identify tab, it makes things a lot easier. And I won't go through it today because it's I'm rapidly running out of time, but there are heaps of really cool keyboard shortcuts as well. So you can actually spend your entire time on this Identify screen without ever using your mouse. All you have to do, you can see that there are actually a whole lot of keyboard shortcuts here. So this is a really easy and quick way of identifying things for people. So the final thing that I'll go into before I jump into the questions is joining projects. And joining projects is one of the most important things that you can do on iNaturalist, because this is often how your observations get seen by people. So projects, for people who aren't aware of them, are basically like a platform nested within iNaturalist that collate like observations. So as a great example that Erica mentioned earlier, here is the Hungry Parrots project. And what the Hungry Parrots project does is it collates observations of parrots feeding on something. So if we click on this one as one example, here is an observation of a sulfur-crested cockatoo. And you can see it is feeding on prunus. So it's feeding on a prunus tree. So you can see that here. So it's feeding on a red cherry blossom. And we now have this data in this project. So we've got an identification for the parrot, it's a cockatoo. We have an identification for the plant it's feeding on, and we now have important data for an interaction between those two different organisms. And you'll see thus far, 44 different species of parrot have been uploaded to the project, all of them with some kind of data on what plant species they are feeding on, or indeed what they are feeding on in general. It doesn't have to be a plant. This is, as I mentioned, a really good way of getting things identified. So the last one I will mention is Australasian fishes. So for anyone that uses iNaturalist, you'll probably be familiar with the Australasian fishes project. And what it does is it collates observations of fishes, sharks, and rays from Australian and New Zealand waters. Now, if you look at all the observations that have been added to this project, it's something like 92% have been identified and a research grade. But if you then look at the observations of fishes from Australia that have not been uploaded to this project, it's something like 47% have been identified. So join as many projects as you can and upload your observations to as many projects as you can. And you name it, there's a project for it, right? If you're interested in fungi, you can join Fungi Map. If you're interested in, for example, there are projects where you can see organisms interacting with plastic. So this is one I set up. So if you find a seagull with a fishing line tangled around its foot, or you see some biofowlers washed up on the beach attached to a plastic object, you can upload it to this project. One really important one is the bushfire recovery project that Libby alluded to earlier. So if you go into an area that was torched by a bushfire, anytime within the past year and a half, you can upload it to this project that myself and my colleagues at the University of New South Wales set up. So what you can basically do is you can go into an area that was burnt by a bushfire and you can record how the ecosystems are recovering. So you can see that over 14,500 observations have already been added to our project, all across the Eastern Australian coast, as well as down into South Australia and Stirling National Park in Western Australia as well. And indeed, for the coming bushfire season, which hopefully will not be too severe, we are also accepting observations into this project. So in the coming spring and summer, if you encounter any bushfires near you, obviously once it's safe to return to those areas, please upload those observations of animals returning to the area or of plants resprouting or reseeding to this project. And finally, before I answer the questions, as Libby also mentioned, we have the Great Southern BioBlitz coming up. So this is an event that will be held from the 22nd to the 25th of October. Hopefully all of the lockdowns across the country have simmered down by then. But this is pretty much a 4-day BioBlitz where you are encouraged to go out and see as many species as you possibly can during those 4 days. You don't have to manually do anything. All you have to do is go out into your area, view, take photos of things, upload them to iNaturalist, and they'll automatically be included in the project as long as they fall within the defined area. And so that should be just within your general South Coast area. So that pretty much wraps things up in terms of what I had planned. So I'm more than happy to go through the chat and look at the questions now. So, alrighty. So Doug posted a great link by Kevin Bottom. And Doug said 6 minutes to go to the Q&A, and that was 20 minutes ago. So I'm sorry for going over time. All right, just scrolling through these questions. So question from Barry, do we always use common names? So you can use either. So you are completely fine to type in common names and they will appear. So if I type in magpie, you'll see that magpie appears as one of the options. But similarly, if I start to type in the scientific name, you'll see that magpie also appears. You can actually change in your profile, which you see first. So if you go to your profile and you click Edit Account Settings and Profile, and then you go to Content and Display, you can actually see how names will be displayed to you across iNaturalist, and you can pick any of these. So you can pick that the common name will be shown first, and then the scientific name in brackets. You can pick the opposite, or you can pick that only scientific names will appear. So unfortunately, there's no option to only see common names, but of course that's because many species don't have a common name. So a lot of obscure bugs and invertebrates. But certainly you can pick which of those you want. And as I mentioned, whether you type in the common name or the scientific name, both options will come up for you. And Annie said, could you explain how to create a species list for a specific location and how to find out if threatened species occur there? Certainly. So if you want to figure out what species occur in a particular area, you just go to that Explore tab. And let's say I type in Greater Sydney. And I'm interested in specifically the land snails that have been seen there. I can type in land snails and slugs. I can now see that there are 32 species. And let's say I'm only interested in the ones that are threatened. All you got to do is click this filters box. And then you see here, there's a little tick box for threatened. You'll see that there are 2 officially labelled threatened species here in Greater Sydney. So one is the Cumberland Plains land snail, and the other is Sauroconcha marshalli, which is a sister species to the Cumberland Plain land snail. So what we might do is we'll actually do birds just so we get more to our list there. Okay, so if we go for birds, we'll see that so far 27 different bird species that are listed as near threatened or above have been seen in the general Sydney area. So to then export that as an actual list, what you do is you'll click filters, and then you'll go download here in the bottom right corner. That will take me to this screen. And what we'll do is this is loading. So this will just take a while to load, but basically what this does is this will load, and then you go down to the bottom and you click create export, and then that will actually create a CSV file for you that you'll be able to download. Now, I will highlight that unfortunately, when you do export data this way, the threatened species filter isn't one that carries over to here. So what you would have to do is you would have to have that those list of 27 species open in that tab, and then you would have to manually add each of these species to that list. But pretending that we aren't specifically looking at threatened species, and let's just say we wanted to look at all bird species in Greater Sydney, and this is a great way for you to export the data to a CSV file, and you've got all these options as well. So you can tick any of these options and they will all come with a CSV file with you as well. So are there any other questions that people have? As I mentioned, tomorrow night from 8:30 to 9:30, I'll be running a kind of 1 hour to 1 hour and a half workshop that will cover similar things that I covered today, but also some more detail about some of these basic uses of iNaturalist. And then on the 7th of September, I'll be running an advanced workshop where I'll be getting into a lot of really nitty-gritty stuff. And then later in September, sometime at the end of the month, I'll be running a second beginners workshop if you weren't able to attend tomorrow's or today's. So, as I said, happy to answer any more last minute questions, or if people don't have questions here, more than happy for people to message me on iNaturalist at any time. I'm the Beachcomber, or to send an email to me. If you do want to ever send a personal message on iNaturalist that is only seen by that person, all you have to do is click this little envelope symbol up here. You click view your inbox, new message, and then you type in the name of the person. Right? And then you can just start typing the message as you would a normal message. So feel free to address it to the Beachcomber at any time, and feel free to message me at any time on iNaturalist if you need any help with anything, or to send me an email.
Great, Thomas, thank you very much. That was fantastic. And as usual, you know, there are quite a number of questions when you go through in so much detail, but your session on September the 18th, was it?
September 7th, I think. I'll have to double check. I think the 7th is the advanced one.
Okay. So,
and the beginners one is tomorrow night.
Okay, so I would encourage people to sign up for those sessions. I think I'll be going away and joining as many projects as I can, because that's a great way to make sure your data is actually used for research, but also it makes it more efficient to having IDs put in place for some of those species. So I think that's a really great tip, and I encourage people to do that. Thanks so much for your time, Thomas. Also, thank you and to your colleagues as well in California for all the support that they've shown, both Libby and Libby's project and Buttawang Coast in transition to iNaturalist. It's really made our life so much easier. So thank you for that. Just another point I would make, Libby mentioned BioBlitzes. Libby's one who wrote the book on BioBlitzes. And so if you go to the BioBlitz hub, if you're thinking of running a BioBlitz, the BioBlitz hub has all the things that you need to know how to run a BioBlitz. I ran one not that long ago, and I used that manual for me to step through what I needed to do, who to get in touch with, all the things to organise. So it was a really fantastic resource. So I encourage you to head over there. All right, I think that's a wrap for today.
Can I just add a couple of quick comments,
yes,
Annie?
Absolutely.
Libby,
Go for it, Doug.
Libby, it's the celebration. I think, did you want to just make a closing comment?
No, I mean, that really, I've only just realised it is 10 years. It was only last week that I realised that we got to some kind of a milestone. So we will be doing other things. I promise after Science Week is finished, we'll be getting all our Atlas guys together and having a proper celebration. So we've been helped, and hundreds of people have contributed a huge amount to where we've got to. But I think now, having moved to the iNaturalist platform, then the future looks fascinating. And I think it's richer, and we can do so much more, and we can do more real science. So I'm delighted that we're here, and encourage everybody to use it and explore it, because it's a treasure trove. For anybody who's a naturalist, there's a treasure trove of information and beautiful photographs and wonderful things to find out about where things are and what's there. So it's all good.
Thanks, Libby. And look, just a couple of housekeeping things. In the chat, I put an evaluation link for Inspiring Australia, Jackie Randall's The manager is always keen to get good data from participants. So if you could fill in that evaluation survey, it should, when the meeting closes, take you to a link in your browser. But if that doesn't work, if you can just click on that link in the chat and fill that out, that would be wonderful. I just quickly wanted to show you a couple of things, because we're very keen for all the people that participate in these events to go and see what other things are on. On the Boonda website, we've got the festival web page, and right at the top, if you click on that link, it'll take you to Libby's page with the Atlas of Life with the remaining events. We've got the Glossies in the Mist and the Gangang Cockatoos tomorrow, Science of Nest Boxes, Echidna CSI, and Forest Monitoring and Improvement Program. Unfortunately, the Life in the Cemeteries live event won't be able to go ahead, but Boonda had about 7 events that were live this week, and we've had to postpone them, and the kids will be disappointed. Everyone's disappointed. On Thursday night, David Howard from Clean Energy for Eternity talking about his home battery system, and I'm going to put in a mention about our electric car. So in terms of applications of science for modern living and reducing your emissions, there's some good information there. We've got a couple of new additions. Jo Lane, who some of you might know from her time with National Parks, is the owner of Sea Health Products. She's doing work with using kelp for a range of health and wellness products, and she's going to do a session on Friday, the 20th of August. And look, I'm really excited about this because Minka Waratah, the Bega High School captain, who is a young lady who interaction with the Atlas, interaction with Boonda programs, she's just got a wonderful enthusiasm for protecting and conserving the marine environment. And her and her dad and Wally, our submersible drone, have done a few trips under the Tathra Wharf. These photos that you can see were taken yesterday by her dad, Luke, and some beautiful images there on the website. And they're just lovely things which will highlight. Now, the website, and they've disappeared for a second there, the website has the link there to register for that webinar. There is work, $7 million of funding for restoration of the Tathra Wharf, which is in itself a wonderful heritage outcome. But Minka is incredibly concerned that the project's going to involve just blasting all these living things off the pylons. So she's keen to get people to help raise awareness of what's actually there and to look at whatever measures can be done to conserve those things. Look, we're trying to keep this as a journal of what's been happening. Wonderful day on Friday at the Civic Centre. Seems even a little bit poignant now that we had all those kids together from different schools. They were separate schools, one school at a time. But the Atlas and volunteers from the library and 2Pi and a whole range of local organisations put on a really good show of science and STEM for them. And all the presentations are posted there. So if you have friends that missed out and you want to highlight some of these things, Bega Cheese on Friday, the wonderful symposium that we had on Saturday, which we had, people like David Booth, Melinda Coleman, Neil Mallon, Navid, all those presentations are there. And because I've been industrious behind the scenes here, we've even got the beaver bet up there already. So no reason to miss out. And there are a whole range of things that were postponed, unfortunately, due to COVID. So thank you all for being here and your wonderful enthusiasm for science. Our community and our civilization relies on it. So, and Thomas, you blew me away there. I've just a bit of a novice with iNaturalist, but I'm really looking forward to getting into it even further. And probably the last thing I would show you is a picture which alludes to what Libby said. That's Paul Whitington with the camera there, and Stuart Harris, of course, the Maratus peacock Spider-Man. And this was at the BioBlitz down at the Bega River. And, you know, it's a wonderful thing to see kids queuing up to, you know, get the expert opinion on what they've collected from these enthusiast citizen science, academics, whoever. I even look at this situation here. I think this kid's trying to push in the one on the left, and he's trying to get to the front of the queue, and the other student's saying, don't do that. So, Libby, I'd just like to thank you again for the work that you and the Atlas have done to help students access wonderful resources and information about biodiversity from the micro-eye digital microscope that you loaned to us and all the other things which are flowing from your work. And it's I guess with the new app, it's really going to take off, I think, because we've got a better platform to do that.
And I think it's just wonderful that we've now got a sister project, and we're interested in the same things, and we can maximize the work over the area. I think that's really exciting. It's wonderful to have friends to work with like that. So it's good.
So thanks, everyone. I'm going to press the button that ends the meeting. Have a great week, and hopefully we'll see you in some of the other activities.
Thank you. Thanks, Libby.
Bye.
Bye.